Hi everybody, Professor Ruggieri here. We are 2 days after we initially inoculated our tryptic soy broth with either Micrococcus luteus or with Serratia marcescens. And what you can see now, and I'm going to compare it to an uninoculated tube. Look at how the uninoculated tube is really clear, and that this is really cloudy, okay? So here, I haven't shaken it or anything. This is the Micrococcus luteus culture from 2 days ago. And you can see on the bottom, it's extra, there's some sediment, it's extra cloudy, okay? And then if I tighten the tube, the cover again, sorry for the police sirens. If I tighten the cover, because remember it was a little bit loose, and I just agitate it ever so slightly, you can see how there's that filmy, that white wispy stuff inside. It's cloudy. That's all the Micrococcus luteus bacteria that has grown. Okay, so I'm gonna put that back over here and do the same thing with the Serratia marcescens culture. What you can see that wasn't present in the Micrococcus luteus culture is this sort of skin on the top. It's called a pellicle. Okay, it's almost like when the fat rises in like an Italian sauce or gravy or soup. And then you can see all the cloudiness and then a bunch of sediment at the bottom, right? And if I tighten the cap and agitate it like this, you can see just how cloudy and turbid it is. And there's some sliminess. So there's definitely been growth. And I want to apologize because it was not clear, and especially since I didn't have the lab kit when some of you got yours. These cultures, these broth cultures, and it's good that I put the date on there because we know when it was started. These are going to be used multiple times in other experiments. If for whatever reason you threw out some of these, then get in touch with me and I can possibly get you some subcultures of these, or we'll figure something out, okay? So if you recall, on Friday we inoculated for both the antiseptic technique and the isolation streak plate technique lab. We are also going to, because it also uses Serratia marcescens, set up for the lysozyme assay or experiment that's slated for this week. Okay, so the antiseptic technique lab uses just the Serratia marcescens. So I'm going to put the Micrococcus luteus back here. And what this lab is all about is going from a broth culture, okay, into a tryptic soy agar slant culture, into a tryptic soy broth culture, right from broth to broth, and then from a broth culture onto a nutrient agar plate. And you'll notice that I always store the plate inverted, or agar side up, bottom side up. So what are we looking for here? Aseptic techniques means without the introduction of contaminants. So you haven't seen this on solid media yet, although some of you did it before I did. And Serratia marcescens, under optimal conditions, it might not be optimal just out in your house, but it should be a pinkish color. So if you see colonies that are maybe any other color, although Serratia can be white too, if you see yellow or orange, or the colonies don't look the same in terms of shape, then you might have gotten contamination. So this just tests your ability to go into the three main types of nutrient agar or broth, right? The slant, the broth, and the plate. So we're gonna light our candle. If it goes out, just keep going. I know that these don't work the best. Oops. Okay, and again, we just want to keep this going and hopefully be able to run the mouth of the tool, or of the tube rather, through there just to maintain sterility. So I'm going to open up my loop, okay, handle side out all the time, never touch the loop. I'm going to draw this out. This is plastic, so I'm not going to flame it, but just put it over the heat, okay? I'm going to go into my tube of Serratia marcescens, make sure the cap is tight, mix it up a little bit so I have equal distribution. Then I'm going to open it up. Okay, pretend like the flame was going. Flame the mouth, go into it, and think of this like a bubble blowing wand. You should see a skin or a film of bacteria on that loop. Okay, put this over here. I know it says to flame the loop again, or the tube, don't worry about that, just put the cap on. And I'm gonna go first from broth into new broth. Okay, so I'm gonna open this up. If my flame was still going, I would run the mouth of the tube through the flame, and then I'm going to put that loop full of bacteria down into the broth, give it a little shake, withdraw my loop, flame the mouth of the tube, cap it almost so it's tight, 1 notch back, and put it here. And then I'm gonna put my loop into the 70% disinfecting solution. Okay, now notice I didn't label this ahead of time. I should have, but I wanna do it right now. This is gonna be S. Marcescens, my initials, KR, and then today's date. So today is 5/31, okay? And I'm gonna put that over here. Okay, so then I need to go from the broth culture into this tryptic soy agar slant. And before I open this up, and I've learned my lesson, right? First I'm gonna label it. So this is gonna be, and I'm just writing SMAR, you'll know what it is. KR, and then 5/31. And I'm gonna open that up a little bit so it's easier when I have my hands occupied. Okay, and I just wanna show you on the slant. The slant has that sort of juicy part. See it at the base? That's actually called the butt of the slant. If at all possible, when you go in with your loop full of bacteria, try not to touch the surface of the slant until you get to that juicy part at the bottom. Stick it in there and then pretend like you're frosting a cake or you're putting peanut butter on like warm bread, just along the surface of the agar. And it's gonna be the same thing when we do our plate. So I'm gonna grab a loop, okay? I'm gonna tighten the tube on the S. Marcescens, mix it up gently a few times. Flame the mouth rather, go down into the tube, grab a loop full of bacteria, flame it, cap it, put it back, open up my slant, flame the mouth. Okay, and I'm gonna go in trying to stay off the surface until I get down to that butt area, and then ever so slightly just slide it in a zigzag fashion right along the surface until you get out of there, okay? Flame the mouth, cap the tube, right? Making sure you're loose 1 notch, put it in here, and then your loop goes in the alcohol. Last thing we have to do is our plate, okay? And it does instruct you just to go in a zigzag motion all across. What I would encourage you to do is then turn it and do the same thing in the other direction, turn it another 90 degrees, zigzag it again, and you'll see what I'm gonna do to try to cover the whole surface. When you label an agar plate, one, you always want to label the bottom, the part with the agar. Why? Because, and you can label both, but if all you label is the lid, right, that lid could easily come off and get confused with the lid of another plate, and then you risk contamination. You also only want to label along the edges. Why? So when you're looking at a plate, maybe under the microscope or under a magnifying glass, the words, the marker doesn't get in the way. Okay, so this is going to be S. Marcescens. I'm just going to write S. Mar. KR, my initials, and then 5/31. Now, one thing I'm going to write on here that I didn't have to write on my broth or my slant was what kind of media it is. Why? Because on here it says it. There's a paper label, tryptic soy agar, tryptic soy broth. Here, this could be anything, right? So this is nutrient agar, so I'm going to write on it NA for nutrient agar. Mind you, it's kept inverted just until the minute where I'm going to streak it. So I'm going to take my loop out. I'm going to take my culture of S. Marcescens, tighten the lid a little bit, agitate it to mix it. I'm going to flame the mouth of the tube. I'm going to go in, get a loop full of bacteria, and it stinks. I'm going to flame the mouth again, put it away, and that's when I'm going to open up my plate, take the bottom out, okay, go in a zigzag motion, very gently, just right across the surface. Notice I'm not gouging it. Turn it 90 degrees. Do that again. Turn it another 90 degrees. Do it again. And then one last time. Okay, I'm gonna put it back inside the lid, keep it upside down, put this in here. Put this aside. Now mind you, this, right, when you do your write-up, is probably where you run the greatest risk of contamination because this has the greatest amount of surface area. So when you open it up and I'm talking and dust is settling, there could be contamination of this, okay? But if the flame had stayed on, and I know many of you have had trouble with this, not your fault. We're not working in a lab, okay? So I'm gonna follow this video with a video of how to do the next lab, the isolation and spread plate technique.